Thank you very much, and welcome everyone who is so far on the line. I can see that the number of the participants is still sort of trickling upwards, but we've just approached the hour, and perhaps we can make a start. Today's session is being recorded, and we're very pleased to welcome those of you joining us live today for our Potency topic webinar. Just before we get into the topic for today then, a few welcomes and introductions to share with you. So today's entitled webinar is Applying a Matrix Approach for Potency Assay Development for In Vivo and Ex Vivo Gene Therapies. My name is Sarah Curry, facilitator in the Cell and Gene Therapy Forum, and I will be hosting today's session that will feature a pre-recorded presentation delivered by Shihua Lin of AstraZeneca and Dominic Hildebrand of Bayer. Followed by that presentation, there will be an opportunity for an interactive Q&A session where your questions on this important topic may be shared with panellists Christina Grigoriadou of AstraZeneca also, Dominic Hildebrand of Bayer, Roland Pack of Roche, and Wyndon Chung of Regenix Bio. Before we do get underway with today's topic then, please allow me to very briefly introduce BioPhorum and the Cell and Gene Therapy Forum, particularly for those of you that may be joining today and are not active BioPhorum members so far. So briefly then, BioPhorum's mission is to create an environment where global pharmaceutical companies are able to come together and collaborate to accelerate the rate of progress for the benefit of all. Via meetings and activities, BioPhorum's team of professional facilitators connects experts like you, bringing you together so that you can identify challenges and collaborate on opportunities to accelerate benefits. Example deliverables come in many forms, but may include benchmarking surveys, discussions, and published articles. And BioPhorum's Cell and Gene Therapy Potency subteam are just one of many teams who have collaborated, discussed, and have produced a paper, and this one will be showcased during today's presentation. Companies participate in BioPhorum via membership, perhaps as a member of one or possibly multiple BioPhorum forums. We currently have 9 active forums shown on this slide, starting with the development group, drug substance, fill finish, information technology, medtech, supply partner, sustainability, which is our newest of these forums, technology road mapping, and of course, cell and gene therapy. The Cell and Gene Therapy Forum was established in 2018 and encompasses all aspects of the CGT product lifecycle, from development to commercialisation and beyond. The member-led programme is organised into currently 7 different workstream topics. These high-level workstreams are outlined on this slide, from commercialisation, CGT manufacture, environmental health and safety and biosafety, high-level analytics, raw materials, regulatory strategy, RNA as a therapeutic and gene editing tool, which is our newest of the workstreams, and validation finally. Working across workstreams and leveraging the cross-forum interactions, we currently have over 800 SMEs, representatives from 44 different member companies, working together to address some of the key challenges that you face within the CGT sector. And so far, with a combined total of 22 publications and presentations, with many more in progress and to come. Within the high-level analytics workstream, BioPhorum members have been collaborating on approaches to determine the potency of cell and gene therapies, an important and complex attribute for these products. In September of last year, members of the BioPhorum Potency team published a paper on the BioPhorum website entitled Challenges for Potency Assay Development for In Vivo and Ex Vivo Gene Therapies and the Matrix Approach. And it's my honour and pleasure to welcome today and introduce some of the members of this team, including Shihua Lin of AstraZeneca and Dominic Hildebrand of Bayer, and Christina Gregorio Du, Roland Pak and Win Chung, who in this webinar session will present an overview of the team's paper, which we hope brings an awareness of some of the challenges associated with developing potency assays that adequately reflect the mechanism of action of gene therapies, and critically, that proposes a matrix approach to potency assay development to aid increased understanding of the complex mechanism of actions. In the team's paper, examples of the matrix approach to potency assay development for in vivo and ex vivo gene therapies is included and will be showcased in the presentation, as well as suggested proposed solutions to overcome the challenges often associated with reference standard and assay controls for potency assays. Following the presentation today, Christina, Dominic, Roland and Win, lead authors of the publication, will be on hand to answer any audience questions. And so we warmly and actively encourage you to raise questions. You can do so using the Teams chat. So during the presentation, if you would like to raise any questions to the team, use the chat to document those. And if you see a question that maybe sort of reflects one that you might like to hear the answer on, you can also like that question with the thumbs up. So the more thumbs up we see against a particular question would suggest that a higher priority may be placed on that question in the time that we do have available for Q&A today. So without further ado then, welcome everyone to today's session. I'm going to hand over now to Nettie, my colleague who will run the presentation. You'll hear next from Dominic and Shihua, and then following that we will return for the live Q&A. So very, very much thanks to Nettie for the presentation. Thank you.
You're welcome.
We're going to start with a presentation, a discussion on potency assay development. And if you go to the next slide, let me shortly introduce who's going to present to you the first sessions. The first part is going to be provided by me. My name is Dominic Hildebrand. I'm working for Bayer in the analytical development and clinical quality control as a senior scientist. The second part is going to be taken over by Shihua Lin, who's director for bioassay development at AstraZeneca in the US. If you go to the next slide, I would really like to emphasize before getting started. First of all, what we're going to show to you is actually the extract of a work that was published as a white paper. And the group that was contributing was much bigger than those parts of the group that you're seeing today. Actually, a large group of more than 10 scientists from major pharmaceutical companies, but also key players in the CGT arena contributed to this paper. Which was actually fostered with respect to the process by the BioPhorum organization. And I would also like to use this opportunity to first of all thank the BioPhorum group for making this possible and for supporting us in the process. And in particular here, Sarah Curie and also Helen Hay for supporting us all of the time and really contributing to a large degree to what we were able to publish here. If you're interested in the whole paper, you can use the link provided below to access the full content. If you want to have further information on the BioPhorum group, please use the backup slides, which will also be provided after this meeting to look at the backup and gather more information. So with that, let's start with the topics. If you go to the next slide, what is this paper about? We are talking basically about the development of potency assays for cell and gene therapies with a focus on gene therapy. Why that? Well, we believe this is a very special topic, and wherever you go, it's a hot topic. And the path to establish a potency assay for final license application is a very stony one and not an easy one. So our idea was really to highlight the difficulties and challenges and also provide a path to solve it in a more robust way, which we refer to as a matrix approach. Today, we're also going to talk about reference standards, which are, of course, to some degree, very related to this topic. Some of the aims of this paper are highlighted below. And so at the end of this presentation, hopefully, you are more aware of some of the complexities associated with potency assay development for gene therapies, and also with terms like mechanism of action and what type of role they play. The idea is also that you understand that there is a staged approach, how you can do the development of potency assays throughout clinical development. We also want to provide some practical examples to not remain too abstract. And at the end, hopefully, that can help you to prepare for licensure application, which hopefully at some stage you will be able to achieve. The next slide gives you a bit of an overview of the content which we are going to provide. So I'm going to start with a bit of a regulatory overview. What are potency assays? Also a bit of what we consider as GMT modalities. So what was the actual scope? And then I will hand over to Shihua, who will talk about the matrix approach. So some type of or some aspect of our solution that we propose, and will also provide you with examples and a short extract regarding reference standards and assay controls for such potency assays. With that, on the next slide, I would like to start with a bit of background. So when we talk about potency assay development for gene therapies, we are talking about two entities here. First of all, this is about ex vivo gene therapies, where you manipulate cells, for example, with retroviruses or lentiviruses, and subsequently apply them to humans, but also recombinant viruses like AAV, which you use then directly in humans. Both of these approaches were in scope of the paper. What they have in common, of course, is that compared to classical biologicals, their manufacturing process and also the therapeutics themselves are much more complex compared to what we used to know from biologicals. What's the key implication? I mean, the key implication is complexity goes along with a lack of classical physicochemical methods that you use to characterize these therapeutics to be sufficient to understand whether at the end of the day the therapeutic will also be functional. Meaning at the end of the day, for these types of therapeutic, you have a very increased relevance of potency assays compared to biologicals, where typically physicochemical and biochemical assays are to large degree sufficient to predict potency. This is not the case, or to a lesser degree the case for these types of therapeutics, which is why potency certainly is one of the most critical attributes for gene therapy development. If you go to the next slide, please. So what is potency? And let's start with some definition as provided by ICH Q6b. Potency is the quantitative measure of biological activity based on the attribute of the product, which is linked to the relevant biological properties. What does that mean? Well, it first of all means the first compound or component of potency assays is the biology of the product. And terms that you typically find and requirements with respect to biology are, first of all, the potency assay you choose needs to be linked to the intended biological effect. Secondly, what you also frequently hear is it needs to reflect the mode of action, meaning, again, it needs to be close to what the product actually does in the patient. And finally, of course, one thing that you need or ideally should show throughout clinical development is that the potency assay readout you identify as being relevant also is correlated to clinical response. However, this is the biological side. If you go to the scheme below, you can actually see that there's, of course, much more that usually is expected from a potency assay, and that's linked to the use of the assay in a clinical control, quality control environment, and later on in a commercial environment. And that's the idea that the assay is not only supposed to reflect biology, but on the other side, ideally also is fast, accurate, precise, specific, easy to handle, rapid, and should be basically a low effort activity that allows you to compress your biology in a very simplified assay. And as you can imagine, it's usually not possible to get all of that at once, but you will have to compromise, and you'll have to see how can I identify something that reflects the biology, but at the other side is also suitable for QC purposes. And that's quite difficult, as you will see. Next slide, please. Potency assay development is a hot topic, and as you can see, both key authorities, the US FDA, but also the EMA, have published since the beginning of the 2000s specific paper or specific guidelines or draft guidelines dealing with potency assay development for gene therapy or cell therapy. And one feature that you can also see is actually that the frequency of publication is increasing, just reflecting the fact that our knowledge and authority knowledge is increasing over time. So what you can actually expect is that the hurdles are increasing and probably exponentially increasing. And what we tell you today may be insufficient actually in one to 2 years already, and depending who you are talking to. So that's also something to keep in mind. And we don't want to go into too much of the detail, maybe just two short extract. On the next slide, you see a short extract from one of the most recent FDA guidelines, the FDA Chemistry, Manufacturing and Control Information for Human Gene Therapy, Investigational New Drug Applications, a guidance for industry from 2020. And what you can actually see if you look at that is that authorities know very well that there is a more challenging part to the development of potency assays compared to biologicals. And you can see that if you look at the lower bullet point, where you actually can read that FDA regulations allow for considerable flexibility in determining the appropriate measurements of potency, and also that the adequacy of potency tests is evaluated on a case-by-case basis. So a lot of flexibility at the beginning, and also the understanding that you need some type of incremental approach. So probably unlike with biologicals, where you may start with a certain potency assay, potentially preclinically already, and at the end of the day, this is also your commercial release assay. This is not the basic anticipation for cell and gene therapy, but there is some anticipation that assays may change over time, and that there's an approach behind to identify the most appropriate assay. Yeah? So meaning there's some flexibility, which allows you to, for example, not validate the assay early on, but use qualified assays, just having lower hurdles for this initial step. However, at the end of the day, and that's the most upper bullet point, for BLA approval, you need to have everything in place, a validated potency assay, as for biologicals as well. So when it comes to degrees of freedom, and if you go to the next slide, what you can see is that you should not feel too much freedom from my perspective. Because as you can see, that's the second bullet point. It is strongly recommended that the development of a suitable potency assay be started as soon as possible. That also for first in human clinical trials, a suitable potency assay should already be in place. And that, for example, if you go below, lowest bullet point, for an FIH trial, the absence of quantitative limits for potency, biological activity, would have to be justified by the applicant. So do not anticipate that even though authorities are aware that it's difficult to have a fully validated assay, that you do not need an approach to assess potency itself. Just the hurdles for validation are to be discussed, and authorities will to some degrees probably accept if you tried a lot, but if it's just still too early to derive clear conclusions. So it's a delicate balance, and probably emphasizing that exchanging with authorities is a very important piece here, and discussing actually your potency assay development approach. If you go to the next slide, please. So this is going to illustrate, and I think we only see part of the graph we saw before. This is supposed to provide you with an idea of how you can actually select the right potency assay. And if you do look at the different clusters of assays that are usually available, for example, for biologicals, starting with physicochemical methods, then you have biochemical methods, cellular assays, animal models, and clinical studies, you can see that if you look at the precision of these methods compared to the biological significance, that there is an inverse relationship, meaning the more biological significance you have and the more complex these assays are, the lesser is going to be your precision. And at the end of the day, in most cases, when you look for potency assays for cell and gene therapy, you will end up in the middle with cellular assays, because they form a nice compromise between biological relevance, but still they are being capable of being executed in a QC environment. While animal-based models, for example, there's something that you can use probably for phase 1 to confirm that your therapeutic is active, but it's going to be difficult to use that for quantitative assay, and certainly not suitable for later stage QC release. So it's supposed to be substituted. On the other side, biochemical and physicochemical methods, it will be very difficult to justify that these have enough biological significance to be used for a potency assay. We then go to the next slide. Let me familiarize you with some of the challenges of the potency assay development and also the differences to biologicals. First of all, it's important to understand that potency assays for gene and cell therapy are usually much more complex, and we're usually not dealing with one mode of action, but with a variety of modes of actions. If you, for example, think about the tumor cells that you inject, it needs to migrate to the tumor, needs to enter the tumor, it interacts with the immune system. So in theory, you can think of a very large variety of modes of actions. And what's the most correct one? Is there one assay that can reflect all of them? Probably not. So quite complex. So usually in vivo models are something that can be available in the beginning of clinical development, but later on, as I highlighted before, you probably have to substitute those. And usually also these complex assays that you sometimes have to use, in particular if you think about stem cell therapeutics, they're not that specific. Signal to noise ratios aren't ideal, quite difficult to handle. Challenges that, for example, come along with autologous therapies are patient-to-patient variability, limited lot sizes, so it's sometimes difficult to really get enough material to do assay development, and also the stability of the material that you need is usually very low and difficult to store. So you do not have an ideal environment actually to do assay development, because you're lacking all types of materials that you would in the ideal world need. And finally, another topic we will touch today, we have a lack of appropriate reference materials in many of these situations, simply due to the fact that a reference standard approach for autologous therapies usually doesn't work out. And there you then have to deal with much broader ranges compared to biologicals, for example. So if you look at that, you can basically say, well, if I want to fulfill classical requirements as highlighted on the right upper side, so quantitative bioassay needs to be quantitative, needs to be stability indicating, needs to be executed in a classical QC environment. How can you do that with all of these additional challenges? Our idea is that you basically need to start with different approaches at the beginning of your clinical development, and we refer to that as a matrix approach in which you have multiple assays as potential candidates for the future. And with that, I would like to hand over to you, Shihua, to continue with more details on that approach.
Well, thank you, Thomas. Thank you for overview. So from now, I'll go into give you a little bit more detail. Later on, I'll give you some kind of example to talk about strategy for potency assay development for gene therapy product. So basically, we want to have like dual testing concept. What I mean by that, first, try to identify potency assay, which is must need for your product control, quality control, and putting in the system, right? So this assay has to be, stability indicating and need for compatibility testing. And of course, need for lot release. So the functionality assay can be as close as possible to MOA, because MOA could be multiple, it could be complex, and sometimes MOA is not fully understood. So that's why we say when you use potency assay for gene therapy product, you want to mimic MOA as much as possible, but cannot be totally same as patient or in vivo condition. So identifies by matrix approach, I will talk about later, and then this assay should be validatable because these assay want to be used for potency release in the QC system. And the second one is MOA understanding and characterization. Right? So because as we mentioned earlier in this talk, MOA could be complex, and may need a number of assays to start with. And some of the assay may not be, for example, in vivo assay may not be suitable for QC test. Then we use as a characterization assay to do some kind of concept study to get information, and to put together. Then we can identify what critical quality attribute has to be controlled. And sometimes we can change characterization assay into lot release assay if it is suitable. And here's an example using AAV as example to tell to show what kind of biological event might happen from the beginning to the end. For example, to analyze the virus or vector, there's assay associated with that, and then once get into cell, they can further get into nuclear and then start making mRNAs and then or peptide, and then even mature protein. And then you can have different assay to monitor this event, but not necessarily every assay is suitable for lot release potency. For example, you can use PCR for strength or infectivity for titer. You can have other like RT-PCR for mRNA, and etc. And if we want to look at the mature protein, sometimes you can use ELISA, use cytokine, or FACS if the cell surface or intracellular, and use functionality assay to look at even reported gene assay or assay for cellular behavior. Here's another example using CAR-T as an ex vivo gene therapy product. Everyone knows you have to modify cell to become your product. This is a CAR-T here, and then the target typically is in oncology field is cancer cell, right? So you look at your protein or your T cell, and then after modification, you look at the activity for this effector T cell, and how can this cell, how strong this cell can kill your target tumor cell. Again, it says you can have multiple assays in different format to look at each quality attribute. For example, ELISA can be used for container ID test. FACS is very important for the cellular behavior, cellular phenotyping. And most important, you want to have functionality associated with your product. And typically you can have either FACS or ELISA if the cytokine involved, at least a few or one cell line cytokine you want to measure. Right? And then lastly, in terms of killing activity, you may use cytotoxicity assay to measure when combined with your product and then target tumor cell to see the functionality of your product. So here is a busy slide. Just basically, we want to talk about, for example, ex vivo, in vivo, ex vivo is CAR-T, something like that, and in vivo is like AAV mediated. So there are so many different delivery vehicles. You don't have to read. If you are interested, you can look at the paper later on. And in terms of material, you're going to deliver to target cell or tissue is most likely DNA, RNA, or mRNA. And in terms of modification, could be random integration or targeted gene editing. Use different approach, right? So typically we talk about target stem cell, T cell, cancer cell, or something similar. The mode of action is gene expression or change cellular behavior, killing your target cell by changing by delivery, gene therapeutics by these two different loops. And let's focus on potency assay, right? So typical potency assay, as I highlight here, is like viral infectivity, transgene expression, and functionality assay, gene copy number, target cell killing, cytokine release, gene editing efficiency. Right? Sometimes the cell surface activation marker also need to be monitored. And then some assays overlapping can be used for either format of your gene therapy product. So we talk about metric approaches. So we use this one figure to outline what we try to say. From preclinical phase 1 until BLA post-licensure, you have a last different stage of fit for purpose potency assay or bioassay development. So the suggestion here is you will start a multiple assay at the very beginning. As you can see here, preclinical phase 1, you have lots of assay to work with. And some of them could be used for characterization, and then later you can identify 1 or 2 or 3 for QC potency lot release. You don't want to wait too late start working assay development, because even you don't have represented material to qualify or validate your assay. My suggestion, with our suggestion, is to start early as much as possible. And as MOA are so complex, so you want to have multiple assay, look at different attributes. Once you identify those critical CQA associated and stability indicating MOA reflective potency assay, and it is validatable, then you put in the lot release. So by measuring multiple attributes, use different type of bioassay, we call it matrix approach. So let's switch to gear a little bit in terms of reference standard and material or assay control. When we talk about potency assay typically have 2 different readouts. One is use relative potency, as most people do for antibody-based, protein-based therapeutic. Everything you compare to reference standard, you come up relative potency, percent relative potency. But for some case, very often in a gene therapy product, may not be able to have relative potency, and then you use absolute value as a readout. For example, titer, you have it's IFU or PPU or per milliliter rather than relative potency percent. But nevertheless, also reference standard is ideal material you want to have for your potency assay. But in reality, sometimes it's very difficult to have reference standard or reference material in place, especially during early product development. So, for example, you have high variability about your starting material. You have a limited size of your batch. You don't have information about stability, and then you don't understand fully about the potential MOA. And the assay itself is very complex and sometimes not QC friendly. And handling procedure also is much difficult, different compared to protein. Protein tend to be very stable. For CAR-T, very likely you have to involve flow, and that is not routinely, it's not a QC-friendly assay, but it is really critical assay, right? So reference standard has to be well characterized, qualified, and stored at appropriate storage conditions. Stability testing has to be done frequent enough so you can understand the stability profile of your reference standard, and you do trending, assay trending analysis. So if it's not possible to establish the reference standard, now you want to consider assay control. What I mean assay control is typically it's not control including the cake. Ideally, assay control is a material where it behaves similarly to your final product. Using similar manufacturing process, if it's not representative, it's fine. I forgot to mention for reference standard, ideally you want to have to be produced using representative manufacturing process. So for assay control, why is it important? Because assay control help you monitor assay performance and lowering assay variability, including sample preparations. And reference standard also need to be well characterized and then testing frequently in terms of stability. Most important, assay control is useful to establish appropriate resistance suitability for your assay. With good assay control can tell you over time whether your assay perform well. Then if assay is performing well, then you can look at your sample data to understand what sample look like over the time manufacturing development. So in summary, potency attribute is one of the most important CQA for gene therapy product. And gene therapy is really complex, it has multiple MOAs. So it's very difficult to have one single assay, just like a protein for potency lot release. You want to have a multiple assay for characterization purpose and for monitors CQA purpose. So our conventional like structure, issue function, those kind of things might not be applicable to gene therapy. You could still see gene by sequence look like normal, but if not properly delivered or stable expressed into a protein, a functional protein, structure may not tell too much. So, well, the main focus for this talk is metrics approach. Basically, you want to have multiple assays start as early as possible. Then you want to identify appropriate potency assay for control, for quality control, and for also to support manufacturing development as well. And on the other hand, you want to have assays to monitor or characterize for characterization purpose, to understand your molecule. Ideally, your bioassay or potency assay you want to link to not only just characterization but also link to in vivo efficacy like animal or clinical human data. With that, I'd like to thank you for team. As this is a team effort. We just come here to present, and a big team. They work together for a long time. I think come up with a very good outline and suggestion for potency development for gene therapy product.
Thank you very much to Shihua and to Dominic for, on behalf of the team, providing that presentation, outlining and highlighting the content of the team's previous publication. I've included in the team's chat a link to that paper that you can download from the BioPhorum website. And now we have an opportunity for any questions that you might have. We're joined by some of the other lead authors of the paper today, Dominic Hildebrand, who you heard from during the presentation, as well as Christina Gregorio-Du, and Win Chung is also on the line. Hopefully we might also have Roland Park as well from Roche. If you'd like to pop your cameras on, Dominic, Christina, Win and Roland, if you're on the line, everyone can see you then. Nice to see you. Keep me
Hello,
company.
everyone.
Hello. And we're using the Teams chat today. If you do have questions, please do be encouraged to drop those into the chat. Look out for those and read them out. We do just at the moment have one question, Dominic, Win and Christina. You might be able to see that as well. For the enzymatic potency assay, which quantification would you suggest? Absolute or relative quantification? Thank you, Vinny, for that question. Dominic, Win, Christina, do you have any suggestions to Vinny on that one?
Sure. So we have dealt with a number of potency methods that include enzymatic readouts, enzymatic activity readouts. And I prefer to do a relative quantification relative to a reference standard. And so I think that's kind of one of the also, I think the considerations there is what reference standard do you use there? Is there a reference standard for the enzymatic activity for your particular product? You might want to measure it against that as well, in addition to your in-house reference standard for your gene therapy product.
Thank you, Win. Dominic, Christina, and I know that Roland's on the line. Do you have anything further to add in support of Win's?
I agree with Wyndon's statement here. I think the preferable approach is to actually do this relative quantification for the enzymatic reaction as well.
Thank you. And thank you very much, Vinny, for the question. We hope that helped you. If you have any other, yeah. Sorry,
I can see also that Roland is also agreeing.
Yes.
I think somehow he has some issues to activate the camera. I don't know if he's able to open the microphone though, so he can be involved as well in the discussion.
I'm just trying to change his participation to presenter. I think Roland's come in under guest rather than his name, so just bear with me and I'm trying to upgrade him
Sure.
now.
Thank you, Antonio.
We have another question just come in, so whilst we're hopefully going to be able to speak with Roland directly as well, I'll read that one out from Sienna. Thank you. Given that a panel of assays may be required to measure potency, can you weigh in, please, on which assays were retained for characterization versus release? Any feedback that you can share, perhaps, that you may have received from health authorities on what was considered most suitable for characterization versus release? Thank you, Sienna. Are any of the panelists able to maybe offer some insights on that one?
So maybe I can talk like from the CAR-T perspective, right? Let's say that initially we can have like this interferon gamma release to look like for potency, and then have all the other assays looking at the cytolytic activity of the cells, looking at the proliferation of the cells, the granulations, or other cytokine secretion as a characterization assays. Then more we collect data, then we can see where there is a better correlation with the clinical outcome, and based on this data decide which of the methods can be then included in the control strategy and proceed with a validation of that potency method.
Yeah, maybe I can add something here. I fully agree with what you were saying, Christina, and maybe in addition, it's also important to understand that, of course, if you look at the area of assays that is available, I mean, typically the degree of coverage when you think of the full mode of action, in particular this term key mode of actions that are supposed to be reflected, many of those assays which may actually be easy to qualify and validate may only cover a very small proportion. So at the end of the day, choosing one of those may not be sufficient. And so I think from a strategic perspective, what you usually start with at early phases of clinical development is rather one assay that is rather full in terms of coverage, while everything else is kept as unofficial at the beginning. So this may also be an important aspect to consider when you design your strategy. So it's of course a bit of science, but also on the other side, it's a question of what's your strategy? How do you fulfill agency needs? And at the end of the day, it's a mixture of considering proportion of the mode of action being covered, feasibility, etc.
And thank you very much. Now I can also join here the discussion, and also not to forget, very often you are faced with, let's say you have fantastic mode of action assay, but you cannot validate it. Or to bring it in such a robust format. So, and then in this is also a very often an sorry, my voice, and selection criteria, which does not remove this corresponding assay, but it moves it maybe in the for your information or the monitoring system, or as extended, that you have this information. And it's always better to have, let's say, a package of assays which have a different angle of perspective to your assay. Or to your mode of action which you define. It depends, of course. It was mentioned with the enzymatic assay. There's maybe difficult to say we cannot show the activity. Whereas, for instance, when you have a membrane protein or a complex or a higher order structure where there's no activity directly related to that, then you have, of course, then a different assays which you cover different aspects. Yeah.
Thank you very much for that. We have a number of questions coming through in the chat, so perhaps to move us along a little, looking at the thumbs up, a question there from Kozak. What kind of mitigation plan is recommended when no potency assay can be developed that is close to the mechanism of action? In other words, what should be the secondary priority to have a robust potency assay if mechanism of action closeness cannot be achieved? Any thoughts on that?
So that's a difficult situation. Maybe it would depend on what clinical phase you're talking about. I think for early phase, certainly acceptable to just look at infectivity and expression if it's a viral vector gene therapy product. And I think that's where, you have to show the attempts to develop assays that reflect more of the biological activity and as you reach later stages. And it could be where you may not be able to, as Roland mentioned, may not be able to validate those mechanism of action assays. But then those could be added to a panel for like extended characterization. So I would say at minimum, infectivity and then expression. But if you can't measure the mechanism or the biological activity, it's going to be really critical that you at least demonstrate that you've made efforts to develop that, and then whatever you have developed, you're going to have to bring that to the regulator and have some kind of discussion with them.
Yeah, I would say this is the last point. I would say I would strongly recommend to reach out and do this type C or the type B meetings with them, and to show you data what you have done so far and why you cannot do it. Let's say that way, and then to look how it's then embedded in your overall control strategy. Potency is always one aspect, early phase, I think it's fine, but for pivotal studies and later for the market success or the access, then it's very crucial that you have really reached out to the regulators, EMA and FDA, or to the others worldwide. So I think it's very important. And then also go not alone as CMC. Go there with your partners from clinics, from preclinics, and speak with them and say why we cannot show it, but we're convinced that you are the sponsor of your program. So so whatever it is, I wish you good luck and good success there in these discussions with the regulators. Yeah.
Yeah, I think what Roland highlights is very important here. I mean, actually, this is different compared to biologicals. It's a more iterative process. And at the end of the day, you can justify many things. But of course, here even less qualified assays in the package that you can really take out of your pockets in interactions with authorities may make a lot of sense. So this is why it's good to have additional characterization efforts. And I mean, one thing I heard also in the past is that it's at some early point in particular, having your pharmacological in vivo model as an addition to confirm each of the batches is active, even if this is a rather non-quantitative approach, is something that can be requested by authorities, but that you can also offer. So it's a bit at the end of the day, it's to some degree a scientific discussion that you will also have. And the outcome really depends on also the amount of efforts that you have included into your preparations and to what degree authorities say this justification is acceptable. And maybe it's even not possible to really at any point develop a mode of action assay. In particular, if you think about very complex therapeutics, obviously, if you ask the question honestly, a small in vitro assay package probably will never cover the full mode of action. So that's the other side of the coin, I would say. So at the end of the day, it's showing the authorities that you did everything you could. And then there is a path forward. But on the other side, it also means you cannot simply go to authorities and say, we don't have a potency assay, we do that later. This is not going to work. It worked earlier on. But in this group, also the feedback we shared was rather that this is viewed more critical. And basically, you should have a qualified potency assay for phase 1 and in your also in your stability studies. That's the requirement.
Thank you very much, and thank you for that question from Kozakra. And hopefully that turn also perhaps touches on the question from Yolani around when would be a good time to shelf a couple of the assays within this matrix approach strategy. There's a number of thumbs up around the question from Savita around reference material selection. Is there a need for qualified reference material? If yes, then is it needed for all phases of development? If the lot of reference material were to change from, say, phase 1 to phase 3, how do we then perform comparability studies between those phases 1 to 3 with reference material lots? Owen, Dominic, Roland, Christina, do you have any suggestions around that approach from Savita?
So for the reference material, right, that should be, and again, it's not like a reference standard, right? It's not like all the other biologics. It should be fully characterized when it is used. So you need to perform, like as any batch or any lot that you need to release, you do all your, whatever is in your control strategy and characterization as well. Should be fully characterized, have like a nice written report about the characteristics of that material. And then when now you are changing from lot to lot, and that is an issue actually for the cell and gene therapy. Unfortunately, this initial lot cannot be like a high number, right? So most likely you're gonna have different lots during your phases. Then you need to do that comparability, looking at the characteristics to compare these two lots between them. Now, the reference material, however, is not, my understanding, it's not using to report like relative potency, right? So it is, and now let me see if I'm confusing now with the assay control. So the assay control is the one that is used for the system stability, understand your method performance and trading data. And for the reference material, I don't remember now if that needs to be for the comparability, what type of data we need to generate to make sure that they are comparable, because now I'm getting a little confused with the assay control use and the reference material. I need to look our last slide there about the definition of the reference material that was added. Let me only double check 1 second. Maybe if somebody from the team can discuss, and then I can step in to complete my sentence there.
In general, I would recommend to consult the other white paper with the reference standard. It's more described, more in detail, and with examples how this can be handled. So these challenges of validation and qualifications.
But I mean,
the article that Roland refers to, just to let folks know, I've included a link just in the chat to that one as well. Go ahead, Dominic, if you.
Yeah, I mean, my feeling would be, honestly, you need a qualified reference standard. I think, of course, the details around a reference standard are a bit different compared to biologicals, but I think the general requirement is a given thing. And in particular, I mean, if you think about, come back to the potency question, you will always have to bridge between processes. And this is something, unlike for biologicals, changes also in terms of potency per amount of your drug is something that's to some degree feasible and acceptable. And there are also well-known examples where actually you bridge between processes where activities increased from one process level to another process level. And that also then means you need a new reference standard, and the reference standard always needs to be qualified and needs to reflect the current production process. So I would say, honestly, this is even much more difficult compared to biologicals, because you also have to bridge then with respect to your potency assay, in particular, if you reference compared to your reference standard. And I know examples where in the potency assay calculations, actually a multiplication factor has been included to really make across the process versions results comparable. So this can be a very complex question, and I can only encourage to work with qualified material. But maybe the others correct me. But my feeling would be there's a significant risk if you do not have that. I would say it's a requirement unless you have a very good justification.
Like a lot of nodding heads from the panelists on that. Thank you, Dominic. With just sort of 5 minutes to go and some remaining questions, it looks as though the Walter's question has a thumbs up at the moment. How would you design an accelerated stability stress study to confirm that a potency test for cell therapy preserved in liquid nitrogen is stability indicating? Would this be a study including multiple short excursions or prolonged storage at warmer temperatures? Is this a question that any of you on the panel today could offer some insights on? Design of an accelerated stability stress study for potency.
So let's say that I don't have like experience right now to do that accelerated stability study for cell therapy. But my thinking there it will be, yes, it's a cryopreserved in liquid nitrogen. So to do this accelerated, maybe have like at room temperature longer time and see what will be the effect. We are doing like in stability studies, at example, where you keep like at room temperature and then you test potency over like 2-4 hours or 6-8 hours, 24 hours. And then maybe by elongate that incubation time at room temperature, maybe you can have these post-degraded cells that you can then see the decrease in the potency as in order to establish these stability indicating characteristics of your potency assay.
And it depends also which temperatures you select. As I think room temperature is by sure about what you take between. No, is it -20? Is it 4 degrees?
Yeah.
And it depends on there you have to reach out to your pharmaceutical guys and to define what is the glass transition point, for instance, where it starts to have an impact. And this maybe you can consider in your design of your stability programs. Very often it's done only the so-called real-time release, not real-time. The real-time release. So what you have a storage temperature, and then the other ones has to be defined. It's not like in biologics where you have already all temperatures defined there, and the heat stress, this 40 degree, for instance. Here the question is, what is then adequate for your product, your specific product and your formulation which you have chosen, and which storage temperature you have chosen. And in this context, I would like to also to refer to vaccines, viral vaccines, whereas for AVV is very often -80 or below, let's say -60. But you know, there are viral vaccines, they have -20 a storage temperature. So there, the design is there already different. So I think this will change and this will be established in the next years. Also in the exchange here, and we have, I think we had these discussions even in BioPhorum. There was a working group, a subgroup, or surveys. So I can also only say, please share whatever you have with your colleagues here. And I think the more we understand, the more we learn, the more we are, let's say, we define then the adequate stability programs with accelerated ones with the different temperatures. But at the moment, I think this is not yet ready to say, listen, this is the holy grail and this is it. And I think even in your case, cell therapy or the ex vivo gene therapy, I think it's even more sensitive compared then to our viral vectors.
And for this, I was thinking like for this ex vivo gene therapy, right, that the cells are cryopreserved in the liquid nitrogen, and then you thaw, and then in the clinic you administrate to the patient, where they are stay at room temperature until it is administrated back to the patient. So in that case, during your process development, you absolutely have these inducibility studies where you can see how long your material can stay at room temperature and is still okay before it's administered back to the patient. So it should be, if you now leave more time, you can start see that drop in the potency, and that can be used to define the stability indicating properties of your assay.
The team, I think, have a couple more questions in the chat. We had allotted an hour for this one. I don't know if there's an opportunity to take one last question, even though we're just a minute over. Perhaps the question from Daniel, if your gene of interest encodes a transcriptional activator, could your potency assay measure the activity of a gene that transcriptional activator upregulates? Thank you for the question, Daniel. Would any of the panelists like to give this one a go?
Yeah, I think that would make sense in terms of showing the biological activity of the product. That would essentially show all of the aspects, the infectivity, expression, and then at least one of the functions of that expressed transgene product. So that seems to me pretty straightforward. That should be acceptable by health authorities.
Thank you, Wynne. Is there any, I guess, talking points any of the panelists would like to sort of finish with, given the sort of constraints on time? Goes without saying my sincere thank you to you all, to the panelists, to Shihua, who's unavailable to join today as well, for him and Dominic's presentation. To everyone who has been able to attend today, thank you very much. We hope you have enjoyed today's session. Thank you everyone for your questions. I think we have managed to cover those through today. If there is anything further, please do contact me by email, and I will be sure to direct your question to the CGT Potency team. Also included in the chat is a link that my colleague Nettie has provided, highlighting the next upcoming webinar, the user stories for IT to support cell and gene therapies. That is on the 27th of April. Again, my warm thanks to everyone for being able to make it along today. Thank you very much, Dominic, Roland, Christina and Wynne, for being able to support the questions. And to Nettie, thank you very much for your support in preparing for today's webinar.